So it's a real pleasure today to be speaking with Associate Professor Mikael Palner, who's based at the University of Southern Denmark in their preclinical imaging core facility. And this is a fun conversation because I actually know Professor Palner from my time at Stanford when we were both postdocs, where he taught me a huge amount about the practical side of PET imaging preclinically in rodents. So yeah, great to be speaking with you again today, Michael.
Thank you. Good to see you, Sam.
Yeah. How are things going in Denmark?
They're great. I've been busy trying to do a lot of imaging experiments with psychedelics and with other compounds as well. So a lot of
Yeah,
pharmacology going on. Yeah.
yeah. I've seen a few of the psychedelics papers that are pretty neat, looking at sort of decoupled metabolic effects of different psychedelics using some of these technologies. So just quickly, what's your lab focused on?
So my lab is really focused on in vivo imaging in rodents, and with in vivo imaging, I mean PET imaging and fMRI or MRI imaging. And I have a long background, as you know, from Stanford and previous in PET imaging, and we've been designing tracers. This is radioactive tracers that you inject in the body, and then you can image them with a positron emission tomography scanner, and then you can visualize it outside of the body without having to do anything invasive into the brain. So you can actually do repeated scans in the same animal.
Yeah, it's a good point. So I think a lot of people will be familiar with EEG in
Mhm.
psychiatry translation. This is being used a lot. MRI, I think most people have heard of at this point, but I think PET is sort of the lesser known hero of in vivo imaging. So what are the benefits of PET versus some of these other technologies? Where is it complementary?
Yeah, so I really think it's less known because you really need to have a cyclotron on site. So you need to produce the radioactivity pretty close to the scanner because we have 2-hour half-life compounds sometimes. So we need to rush it a little bit. But what it can really do is that it can do molecular imaging in a different way from EEG, that is just electric signals, and MRI, which can do a lot of cool stuff as well, but not molecules per se. So in PET imaging, you will have a molecule that you label with a radioactive tracer. And that can be fluorine 18, for example, or carbon 11. And these radio tracers will then be injected into the body. And using the camera, uh, it can detect the positrons or photons that are emitted from the positron electron annihilation. And that way we can track the compounds around the body. So you can basically image whatever you can imagine is going on. So that can be ligands that couple to receptors. It can be metabolization from sugar metabolization, or it can be, as you and I were working on sigma receptors, where we injected a compound, FTC 146, I think it was called, to bind to the sigma receptors. So basically any biological process in the body that can be targeted with a molecule can be imaged.
Mm. Yeah, so it's got some advantages over things like MRI in that you can track the actual labeled tracer in real time for as long as the scan is conducted and as long as there's radioactivity being emitted, right?
Yes, and
Um,
fully quantitative. So we always
yeah.
we always know how much we have at any given time. So you can do departmental modeling. You can see how much is actually going to the brain and lungs and heart compared to other parts of the body. And you can then calculate binding potentials, which
Mhm.
are closely related to kinetics that are known from in vitro, right? You can simulate stuff from KD to be similar to the binding potential and stuff
Mhm.
like that.
Yeah, so thinking about, psychedelic drug action or even psychiatric drugs more generally, from MRI, you're essentially looking at a downstream effect of the drug on things like blood flow and metabolism and energy use, right? Whereas this can look
Mhm.
at the exact drug and what it's doing in any relevant compartment of the
Yeah,
body.
you could either you could label the drug with a radioactive compound, or you could label sugar, for example, and then you can look at sugar uptake in the brain that the compound is causing as an indirect measure of brain activity.
Right.
Or
And
you
it,
can,
like in an
yeah, or you,
FDG scan, right? In oncology, that's where it's
yes,
widely used because
yeah,
there's big differences in sugar metabolism. But I think beyond that,
exactly.
I think people aren't as aware that beyond outside of oncology, you can use this for studying all sorts of drug processes in a really handy
Yeah,
quantitative way.
yeah, similar to in vitro studies where you measure Ki, you can actually, so you can have a radio labeled drug that could be MDL to binding to the serotonin 2A receptor, right? So if we label that with a fluorine, we have a very good compound And then you can actually inject it in the body. You can see the distribution of 5-HT2 receptors in the brain. And then in a similar way to what you do in vitro when you measure Ki, you can inject a psychedelic drug that could be psilocybin, LSD, or something similar. And then you can actually measure the occupancy or the displacement of the radioactive compound in real time. So, and you can do it in different ways. You can either scan before you give the drug and then give the drug and then scan again, or doing a bolus infusion study where you actually displace the tracer while the scan is going on. So there's kind of different ways, but the main thing is that you have a live animal. You don't take out the brain, you look inside the brain.
Yeah, I'd say Denmark is becoming pretty famous for being sort of world leaders in this particular field for looking at 5-HT receptors as well, right? So you can obviously use sort of 5-HT2A antagonists and displacement there to look at occupancy. But I think you were involved in some of this work, maybe in collaboration with Gitte Knudsen, I think it was, looking at actually labeling 5-HT2A agonists, right? So things that are very closely related to psychedelic drugs.
Yeah, definitely. This was actually part of my PhD project back in the day, together with Mathias Hertz, who is a radiochemist, and then Gitte Knudsen. And it's actually a fun story. I was at a radiochemistry course in Tours in France, and then I met up with Matthias, who was from Germany, and I was doing in vitro studies on 2A receptors, and he needed somebody to measure the binding affinity of his new PET tracer for the 2A receptor. And then I said, yeah, I can do it. Just ship me some compound. And then he came up and we did the experiment. And from there, we just boomed into to producing PET tracers for 5-HT2A receptors, and have since the most known is probably SIMP36 that is used in humans. If you have seen these pictures of occupancy of psilocybin in the human brain, and now also with LSD. And similar, we can do the same thing in rats, where I took my research, and we can do occupancy in vivo in rats using another compound that is called 18F-MHMZ, which is Matthias Hertz compound number something, like a real chemist that names it as initials.
Yeah, I'm familiar with this naming convention. In a technical sense, this is something we've been exploring a little bit, is given that non-hallucinogenic 5-HT2A agonists have these effects that may be sort of a Cmax driven in the same way that psychedelics are there any differences between using a radio labeled 5-HT2A antagonist versus agonist in those kinds of displacement studies?
There might be, but the truth is that nobody has really looked at it with the 5-HT2A receptor yet. But if you look at the receptor models, if you have an agonist, it should bind to the active part of the receptors, right? The ones that are coupled to G proteins or whatnot on the intracellular side. And the antagonist should bind to whatever receptor is available. But nobody has really done the head-to-head comparison of occupancy with two different PET tracers and the same dose of drug yet. Mainly because I think we haven't done it because SIMP36, which is used in humans, doesn't really work well in rats.
Oh, interesting.
Yeah.
Are there reasons for that? Is it understood why it's different?
It's because it's a substrate of the PGP transporter. So there's a efflux transporter in the rat brain that just vacuums everything out of the brain very fast.
Right. Okay.
And it's a real hurdle to get past that when you're doing rat brain imaging and mice brain imaging.
Yes, species differences for the 5-HT2A receptor are also pretty well described, but yeah, I hadn't really considered for some of these traces that efflux effects might be significant there. And there are, to the credit of the team in Denmark, there are relatively few labeled 2A agonist traces, right? SIMP36 is one of a handful of sort of related compounds. But are you aware of any plans to develop, new traces that are maybe in a different sort of chemical family that are not subject to efflux? Do you know anyone who's working on that?
So in the human brain, it's not a big deal. So it works fine for the SIMP36 agonist in humans, but we haven't made anyone that works very well in rats yet. And I don't think the plans are to develop new ones in rats because MHMZ, which is an antagonist, it's MDL with fluorine label on it. It works so well and it produces really good images of the 5-HT2A receptor. So if we are going to make an agonist, we would have to battle first the PGP efflux transporter, then also normal metabolism, selectivity, non-specific binding, and it's a big project to produce a new radio tracer.
Yeah, yeah, I'm familiar with that from our time at Stanford. I remember you visiting the cyclotron that we were working in
Yeah.
underground there as we were radio labeling these things. Very, very cool science, but definitely very involved and not trivial. Very cool. And you had a paper recently that was about some of the metabolic differences with psychedelics. I was wondering if you could just tell us about some of your most recent science in the space and just give us a sort of high-level summary of the coolest findings from that paper.
Yeah, so that's a different way of using PET imaging, right? So here then we use radioactive sugar, same as is used for cancer research all over the world. And then you can study the metabolism of the brain as a proxy for brain activity, right? Because neurons take up sugar and this tracer is then trapped inside the neurons. So if you give a drug and you give the radio tracer, you can actually have it go to the brain over time while the drug is working, and then it gets trapped inside the brain, and you can then do a snapshot of whatever activity happened in the brain afterwards. So after the uptake phase and the drug action, then you can have a snapshot of what happens in the brain. And we have been using that technique to study metabolism changes following psilocybin, LSD, and 2CB in the rat brain. And here we then scan the animals at baseline. We give them drug and scan them acutely, and then we wait 1 week and we scan them again without any drug action. And then we can actually see differences in the metabolization of FGG in different brain regions. Acutely, we have differences between the drugs, but also long-term, following 1 week, we have differences between the drugs.
Yeah, that was a super interesting takeaway for me as we look at a sort of non-hallucinogenic 5-HT2A agonist. We find that there are also differences. There are acute effects and post-acute effects, and these are often a little bit different in terms of behavior and other things that are going on in rodents. So
that makes sense, right? Yeah.
Yeah, what were the biggest differences in the acute differences between the drugs versus the sort of post-acute effects that you're seeing in metabolism?
Yeah, I think the biggest difference was with psilocybin. We found acute effects in the orbitofrontal cortex of rats. And that was pretty interesting to us. There was a lot of different activities going on, but they were then gone in long term.
Interesting.
While we had some thalamocortical connections that were changed, but they were still there long term. So there was something going on in the orbitofrontal cortex that didn't stay for a long time.
Interesting.
And without having coupled it directly, we also know that obsessive compulsive disorder animal models, they have something going on in the orbitofrontal cortex. And it seems like psilocybin is working on these animal models to reduce the symptoms of compulsivity, but they also need to be redosed. So it's not a cure, like in depression, we have animal models that are cured of depression when you give them psilocybin. It's not the same case with these obsessive compulsive models. So you need to redose, and that might be why. So we have an effect in the orbitofrontal cortex that then disappears. So you need to redose in order to get the same effect.
Yeah, that's super interesting. I had a very good friend from high school actually who sadly took his own life in our final year of high school, who had depression and OCD, and he shared with me that he was using mushrooms even back then to very effectively treat the symptoms of compulsivity in his condition, which is pretty interesting. So I think there's been a lot of renewed interest in the preclinical translation, and obviously we don't have very good preclinical models for OCD, but I think there's a renewed interest there through this sort of back translation from various patient populations who are having good effects. Is this something that you saw, do you see a similar sort of profile for LSD or 2CB, or were these totally different?
No, they are much more alike than psilocybin, but both of them are different from psilocybin. With 2CB and LSD, we see a big effect in the VTA and the dorsal striatum. We believe that it might be some kind of dopaminergic action without knowing exactly what goes on. The way I see my research is often that I look at the whole brain and I try to pinpoint some regions that stuff is happening in, and then I hope that other researchers will take our research and then go in and punch in some fibers and then look at what is actually going on a cellular level. So
Yeah, and I'm sure that will be the case. I feel like it's, every week in this field, at least neuroscience more generally, there's huge discoveries being made, but it takes, a lot of curious researchers to really delve into a lot of the details here.
Yeah,
Um,
definitely.
yeah, yes, super exciting.
Yeah, maybe one of the things that we didn't talk about was that this technique, together with EEG and MRI, are really some of the techniques that translates the best to humans because
Right.
we have a human PET scanner, we have a animal PET scanner. So these are really, really good methods to translate
Yeah.
stuff from animals to humans.
Yeah, for sure. I mean, the things that are appreciated for both EEG and fMRI are that the rodent models are generally quite predictive in the clinic, and we can measure these same signals in humans. But yeah, this is also true for PET, right? So there are both
Mhm.
preclinical and clinical PET scanners. So what are the main sort of, I guess for psychedelics at least, I'm guessing most of the focus has been on target occupancy, looking at what dose preclinically and clinically is efficacious based on a given dose of psilocybin, say translating to a particular threshold occupancy for 2A receptors. Is that the right way to think about it?
Yeah, that's one of the main things with the psychedelics at the moment, yes. But I mean, the dopamine field has been uh, really prominent in this way to prime dopamine action because they have some really good tracers to study, yeah, the release of dopamine in humans,
Mhm.
doing all different kinds of things. So I guess that the serotonergic community as a whole at some point also will move in that direction and be able to study some things in vivo.
Yeah, yeah, it's a good point actually. For many dopaminergic drugs, at least, some of these are a little bit more selective for different subtypes. But for psychedelics, one of the interesting challenges is they're polypharmacological in nature,
Mhm.
right? So interactions are also very prominent with 5-HT1A, 5-HT2C, some of these other receptors. Are there good radio tracers for many of those 5-HT receptor subtypes as well?
Quite a few of them, at least 1A. There's a
Yeah.
really good one. We have for 5-HT4 as well, I know.
Mhm.
Um, we have been tried for many years to make one for the 5-HT7 receptor, but
Right.
yeah, so I have a lot of papers with radio tracers that didn't work for the 5-HT7 receptor.
Well, that's the best way to find one that does work. Someone
Yeah,
has to make a bunch of them and try them out, even if they don't
yeah.
work. In terms of sort of therapeutic for psychedelics, do you think, in your scientific opinion, do you think most of the activity, based on what you can see from PET studies, is sort of been driven by this 5-HT2A occupancy and activation, or do you think the other receptors are playing
No.
a super critical role as well? Opinion only.
Uh,
You won't be held accountable
yeah, that's
for
the
your
big
opinion.
question, right? I think there's many receptors that are doing the job. So 5-HT2A is obviously one of them. It it's coupled to GQ and you get activation in the prefrontal cortex. But other receptors are also coupled to GQ. So I can't see why another receptor that is also GQ coupled wouldn't do the same thing. If it was sitting in in a similar neuron, it would definitely also increase plasticity, right? So it's a combination of all of them.
Right.
Of course, 5-HT1A is also important in some way, especially for DMT,
Mhm.
um, because you can see if you shut down some pathways while you increase plasticity in others, of course it will change the whole change of plasticity. But
Right.
yeah, maybe that there's actually another PET tracer that we didn't talk about so much that can image synaptic plasticity.
SV2A.
Yeah, so
Yeah.
it's debated, but it's the best one we have for imaging plasticity in humans
Right.
and rodents in vivo. But
Yeah.
that is a possibility, and
I,
we're
I
working on that right now, so as well.
yeah, I've started to do a bit of a deeper dive on that one, how translationally relevant it is. But I saw that Emmanuel Schindler, for example, in her sort of migraine and headache disorder trials, is now using PET in the clinic to study how much of an effect SV2A might be correlating with in terms of therapeutic outcomes in some of these disorders, being treated
That
with
would be great to
psilocybin.
see. Yeah,
Yeah, yeah, it's a pretty interesting approach, and I hope to see more people sort of using it clinically as the sort of best structural plasticity marker that we have for
yeah.
PET that can be quantified.
And
That's a
then
good point.
combine it with EEG and MRI, right? Because
Yeah,
they're not competitive. They are like if you combine them, you actually get different measurements of biology, so you can really get some good data out of that.
yeah, absolutely. I think it's not that there's a single tool that's superior to everything else. They're actually extremely complementary, which
Yeah.
is what I really liked about your PET MRI work with the psychedelics. You get a lot of richer sampling of information for sure. Awesome. Well, thanks so much for your time, Michael.
Mm.
Always great to speak with you.